monthly seminar. My name is Tony Latai. I'm the president of the SFPM. Before I introduce today's speaker, I would just like to hopefully remind everyone of what I consider to be a very important meeting coming up. Hopefully you can see my screen. This is the EHA SFPM meeting. It's being held in Copenhagen, Denmark in September. You can see the dates 25 to 27, 2024. I consider this very likely to be something of a landmark meeting. It is the first meeting of its kind where we are bringing together really outstanding experts, both in the field of functional precision medicine as well as more standard genomic precision medicine, something that I think is essential for the progress of our field, as we need to make contact with the other people doing precision medicine. And I think it'll help greatly for people to have better awareness of functional precision medicine. I think sometimes some of the barriers we face are not skepticism, but just ignorance of what we're doing. And this meeting will be essential to provide that. So I encourage you to attend, and I encourage you to, if you have trainees, to bring them to attend. Members of the SFPM will get a discount on registration for this meeting, and there will be some travel awards going out to trainees. So I will also use this opportunity to remind anyone who is on this call right now, if you are not currently an active member of SFPM, please join. And to remind you, if you're a trainee, you can join for free. Doesn't cost you anything, but we need to register you as a member. So I'd encourage you, if you are a member and supervise trainees, please get all your trainees to join too. It'd be great for us to bulk up our membership. At this point, I would like to introduce our speaker today. Our speaker today is Russ Jenkins. Russ Jenkins is a colleague of mine in the greater Harvard system, but he is currently on the faculty at Massachusetts General Hospital, and he conducted his postdoctoral research in David Barbee's lab at Dana-Farber Cancer Institute, where he made use of ex vivo studies of patient samples to study response to PD-1-based therapies. He's been on the faculty as an independent investigator since 2018 at Mass General Hospital Cancer Center, and he specializes in investigation of melanoma. He is the inaugural Tumor Early Career Investigator in Systems Pharmacology and a member of the Laboratory of Systems Pharmacology at Harvard Medical School. He's also an associate member of the cancer program at the Broad Institute at MIT and Harvard. So I look forward to hearing from Russ today I imagine it's going to involve prediction of immunotherapies using patient samples, but we will see what unfolds. Take it away, Russ.
Thank you, Tony, and thanks everyone for joining. I'm excited to be here. I've been a long believer in and proponent of functional precision medicine, even before I knew that's what it was called. So thank you, Tony, for kind of leading the field in that direction. So the background on the title of the Living Tumor Biopsies is because the title that I've given, which I'll describe later, was deemed too complicated by some laypersons. But I think that's one area that we can think about is actually how we communicate the science that we do, and I'll bring that up a little bit later on. So let's talk about what we can do with patient tumor specimens to learn how to make better cancer treatments. So these are my disclosures here. The only one pertinent is that during my time at Dana-Farber, Dave Barbee and Cloud Pellets and I spun out a biotech, Xfera. It's in the Seaport, and they do functional precision medicine for industry partners. So I'm a melanoma medical oncologist 1 day a week, and so this plot is pretty important to me, and I think about it pretty frequently. And it's really a landmark study when it comes to cancer immunotherapy for all solid tumors, but for melanoma in particular. And this is the Checkmate 067 study, which made the point that while we've made progress in terms of outcomes for patients in the last decade, we still have room for improvement. But if we go back in time to 2010, if you had a diagnosis of stage 4 melanoma, you had a less than 5% chance of being alive. 5 years after that, on comes ipilimumab, CTLA-4 blockade, and it's maybe about a quarter of patients. And then with anti-PD-1 in the mid-20 teens, we were getting up to the 40-45% range. And with the combination of those two approaches, we're getting close to 50% of patients in some instances more than 50% of patients alive, which is pretty remarkable. But the patients that I care about in addition to these folks are the ones for whom our current treatments do not work. And hopefully we can learn from melanoma and what makes our immune system tick to make better cancer treatments and develop novel cancer therapeutic strategies for other cancer types as well. But after the first run of immunotherapy, things don't work well, meaning that the response to a given treatment after a patient has been treated with a frontline PD-1 or PD-L1 blockade, the outcomes aren't so good. So the tumor has potentially gotten smarter, the immune system has evolved, and for patients treated upfront with PD-1 blockade, the likelihood that you get any real mileage out of combining PD-1 and CTLA-4 is just not that much. It's maybe a third of patients. And the long-term benefits just really aren't there. So the overall response rate was less than 30% compared to, maybe 50%, compared to less than 10% with CTLA-4 alone. And if we take the latest and greatest in melanoma treatment, adoptive T-cell therapy, which has been around in Europe a little bit longer, it's been around in the States at National Cancer Institute with Steven Rosenberg for 40 years, but is now FDA approved as lifileucel, also has a response rate in the PD-1 refractory setting of about 30%, which again leaves a lot of patients with a need for improvement. And in the interest of time, I'll keep moving along. So cancer immunotherapy can provide these amazing, durable, long-lasting responses that look for all the world-like cures, but currently benefits a minority of patients if we look beyond the melanoma space. So if we look at all solid tumor indications, it's just under 50% of patients that are actually eligible for some form of immunotherapy, but those that are actually getting the benefit is a small sliver of that particular pie. So we need to do better in melanoma. We need to do better more broadly. And the market continues to grow. It's expected to continue to grow moving forward. And most of the approaches that are being trialed are combination strategies. In this particular slide, I've shown for years, and it's now getting a little dated. But this is meant to look across all disease indications here. And you can just focus on melanoma. But the patterns here are similar. There's few treatments that end up with FDA approval and land in the middle of the bullseye, and then moving outwards in concentric circles. You've got your phase 3, phase 2, phase 1. So most of the trials, most of the action is in the phase 1 setting, but we know it's probably 90% of those trials that are going to fail. So how can we do a better job of identifying things that are actually going to make it and maybe pull back some of these strategies that ultimately aren't going to reach or benefit patients? So over 1,000 clinical trials evaluating combination approaches, many with an immunotherapeutic partner. And this is from a review of just a couple years ago, looking at how this landscape has changed over the last 6-7 years. Essentially, right before PD-1 was approved, we had a few combination approaches relative to monotherapy approaches. But after PD-1 became approved, all of a sudden there was a huge expansion of these combination trials building off of PD-1 or PD-L1 backbone, and that has continued as time has gone on. And there's a really lovely review that was written a couple years ago, essentially taking stock of where we've come and the successes and the failures. And it was the top 10 challenges facing cancer immunotherapists or in cancer immunotherapy. And one of the top ones has to do with the development of preclinical models that translate to human immunity. And the notion there is that humans are indeed not mice. There's a lot of major differences between the established immune-competent mouse models and patients, and even in between patients, we can't capture the heterogeneity that we observe in the clinic with inbred syngeneic mouse models. And this is something that would be ultimately beneficial both to the industry folks that are making these drugs and academic medical centers where we're treating these patients. And this could be deployed at multiple stages of the drug development life cycle, reducing the failure rate by identifying things that aren't going to work earlier and essentially reprioritizing things that have a better chance. Maybe learning about biomarkers of sensitivity, resistance, de-risking some of those emerging targets, and as I mentioned earlier, kind of deprioritizing those ineffective strategies earlier to make way for things that maybe have a little bit more promise. So we think in several aspects of the drug development life cycle, this could be particularly impactful. So our solution to this are patient-derived organotypic tumor spheroids. They're frequently referred to as organoids, but in the strict Hans Cleaver sense of the term, we don't qualify. Organotypic implies 2 or more cell types grown in a 3-dimensional structure meant to recapitulate normal human physiology. So I think what we're aiming for better fits that description. Again, I'll provide a little bit more of a background, but essentially the idea is we take a tumor from a patient, we dissociate it, we disaggregate it, and we embed it in a microfluidic culture device in 3D in a collagen hydrogel, although other extracellular matrices can be used, and then we can flow in, add different drugs, antibodies, even cell therapies, and study in a patient-specific manner how a given treatment works. And we initially published this. This was work from my postdoc in the Barbie lab at Dana-Farber, and we've subsequently published a couple other studies since I started my own independent lab, and the Barbie lab has continued to use this. This is just a quick fluorescence photomicrograph showing what it looks like under the microscope, where you have a bunch of tumor cells here in green, with a bunch of different types of immune cells all embedded in. So essentially, whatever the tumor infiltrating immune contexture is in that tumor can be taken outside of the living organism and studied for these short-term ex vivo cultures to study the dynamic interactions between tumor cells and immune cells. So the best model of human cancer, we think, continues to be human cancer. And this is taken from a review from Calvin Kuo and his group, showing ours as one of several different approaches to do this. And this is just a nice simplistic workflow, showing what the process is from going to digested tumor fragments, to tumor spheroids, to microfluidic culture, to having these multicellular tumor aggregates in this microfluidic device, and all the various downstream analytics that we can bring to bear. And this is a cartoon of what it looks like here, which hopefully will run on its own. But you've got in the center gel region here, your tumor spheroids. You've got these anti-parallel media ports on either side. And using a standard pipette, you can introduce small molecules, antibodies, you name it, and then study over a limited time window what transpires, whether or not there's more killing of tumor cells, whether or not there's alterations in the immune cell contexture, or in secretion of inflammatory cytokines. So what does it actually look like? So on the left here is a video from a former patient of mine who had melanoma that had metastasized to the brain. Despite receiving immunotherapy, so this was an immunotherapy resistant patient, the tumor was explanted, we received some of that material, and this is what it looks like on the individual spheroid, time lapse over 24 hours. So you can see that it's not just a static blob, it's moving, it's dynamic, and you can see these kind of finger-like projections moving into the collagen over a relatively short period of time, again, just 24 hours. And this patient ultimately recurred at the surgical site and required another neurosurgical intervention 3 months later. Now, in contrast, on the right, we have a tumor spheroid from a patient who was responding to immunotherapy, but had a residual lymph node under the arm that was bright on FDG PET scan, so it was removed. There's a modest amount of residual viable tumor, but we can see the picture is very different here. We have these small round cells dancing all over the place, and it looks like they're sampling the exterior of the tumor spheroid here, and ultimately there is not the same degree of growth. So this, for us, looked for all the world like we were having immune cells dancing around in the collagen matrix and keeping that tumor from growing. But ultimately, we want to do more quantitative analyses than just these pretty movies. So we've demonstrated that all the different spheroid fractions we use actually retain the same immune cell populations using flow cytometry. We can do collect images using standard light microscopy at baseline and 3 days and 6 days and watch the evolution of the tumor spheroids over time. We can do single cell RNA sequencing, although sparingly because of the cost. And we can do cytokine sampling of the conditioned media in the microfluidic system to see how the cytokines that are secreted change over time. We can do limited scale multiplexed immunofluorescence, just 3 or 4 colors at the moment. We're working to enhance that. And then we've also done some bulk RNA sequencing as part of a separate effort looking at resistance profiling. Our standard workflow is something like this, which we initially developed and using syngeneic murine models. And here is the MC38 syngeneic model, which is a stereotypically PD-1 sensitive model where we took tumors from mice that were treatment naive, embedded them in parallel microfluidic devices. Some received an isotype control, others received the anti-PD-1 antibody, and then we did a fluorescence-based viability assessment using acridine orange and propydium iodide. We could see after 6 days of treatment with an anti-PD-1 antibody, we actually saw a very nice and robust increase in the dead cell signal. This deconvolution fluorescence microscopy showing that at the level of an individual tumor spheroid, and contrasted that with a tumor spheroid from the B16F10 murine melanoma model, which is a stereotypically PD-1 resistant model. And we've turned the crank on a number of different models over the last several years. And again, there's a nice general concordance between whether or not there's thought to be some in vivo sensitivity to a particular model system versus PD-1 resistance in terms of the in vivo versus ex vivo characteristics. And again, differences in the cytokine profiles that mirror those different sensitivities that we've seen. And I'll move quickly through this since this is all published several years ago, but just to provide the background to those that are a little less familiar as to the testing that went into this. But we can even go to some of these models that are heterogeneous responders. Some of the mice respond, some of the mice don't. And you can actually challenge the mice in vivo now with an anti-PD-1 antibody and get tumors that have regressed and have stabilized. These are the responders here in blue, or those that have progressed, the non-responders here in pink and orange. And we can take those tumors, make spheroids out of them, and then challenge those spheroids either from responding or non-responding mice and see whether or not they preserve those characteristics, those ex vivo features of sensitivity. And indeed they do. The responders can be re-challenged in the microfluidic device with PD-1 and we get killing, whereas the non-responders do not. So this was a nice experiment. We've collaborated with a number of different folks for the last several years to test novel combination strategies. This was taken from a companion paper that was also in Cancer Discovery with Nathaniel Gray and Kwok Wong's group looking at CDK4/6 inhibitors. And these inhibitors cause kind of cell cycle arrest and are not cytotoxic on their own, but they mean they will ultimately make the tumor spheroids smaller so they don't grow quite as well. The PD-1 antibodies in the models we tested killed a little bit, and the combination, you get kind of a hybrid of those two. So smaller spheroids and a bit more death. And we've done this in the murine model, the MC38 I mentioned before, and tested a couple different CDK4/6 inhibitors, including trilaciclib, which was developed by Nathaniel Gray. And again, in the interest of time, I'm going to keep moving to get to the major part of the story that I wanted to get to, the new, as yet unpublished data. But the three big things we work on in the lab, first is understanding response and resistance, the PD-1 blockade. The second is trying to develop rational combination strategies to overcome resistance. And the third is to try to continue to hone our skills at studying these living tumor biopsies or PDOTs or MDOTs outside of living organisms and doing some of the tech dev work. And the long-term vision, we hope, is that eventually, potentially in combination with static biomarkers, that we can get a little bit smarter about how we decide what treatments an individual patient should get, appreciating that patients that progress on multiple lines of treatment often have maybe only one or two shots on goal, as we say. So being able to pick the most appropriate and most effective treatment is likely the best way we can benefit these patients. So one way in which we've identified drugs to partner with, let's say, an anti-PD-1 antibody has been doing CRISPR screens. And I'm going to condense a lot of story that, since it's all published now and those that are interested can read it, or you can reach out by email. But we had identified a particular kinase, tank binding kinase 1, as a potential tumor intrinsic immune evasion gene using a whole genome in vitro CRISPR screen. And others had nominated this target as well. We had an inhibitor. We said, let's go ahead and do this experiment. So now we're going to run not just a single PDOT specimen, the patient version of this, but we're going to run a phase -1 clinical trial where we actually test this combination strategy of an untreated control, an anti-PD-1 antibody, here pembrolizumab, a TDK1 inhibitor provided by Gilead, and the combination of these two. And we're going to let them run for 5 to 7 days and then perform our fluorescence-based viability assessment at the end. And in this particular cohort, there's different tumor types here. These are the different colors, which aren't annotated, but just for reference, the teal color here is DNA mismatch repair deficient, or MSI high colon. And if you see the bars going down, that means there was more killing. If the bar is going up, there was tumor growth. Some of the MSI high cancers here are fairly responsive, which fits with the clinical scenario. The yellow or saffron color here is renal cell carcinoma, and black is melanoma. And the pink here is triple negative breast cancer. So some of the stereotypically more PD-1 sensitive tumor types. But there's plenty of black melanoma specimens that did not respond in this particular cohort. And overall, we didn't see anything that was statistically different if you look at the cohort in its entirety. Whereas the TDK1 inhibitor did have some single agent activity, a little bit more prominent than the anti-PD-1 antibody, but it was the combination of the two that made the biggest difference going forward, with deeper responses and more patients responding than with either of the single agent treatments alone. So pivoting to the second half of the story about kind of the newer unpublished data, the other strategy that we've developed to try to nominate targets isn't just things like an unbiased whole genome CRISPR screen, but given the advent of single cell RNA sequencing, trying to leverage this single cell resolution gene expression data to nominate targets, and then bring those, try to identify things that are either targetable or for which targets already exist, and then bring them forward and test them in our PDOTS model. So our next adventure is a cell surface target called CD38, which I'll tell you a little bit more about in a moment. But this was a project, I'm going to start off with some of the credits now, led by a postdoc in my lab, Orion Revach, who's going to be on the job market soon. A very talented technician, Angelina Cicercia, who's now a first-year medical student, Moshe Saad Feldman and Nir Hacohen here at MGH in the Broad. And then a cast of characters has been just a real privilege to work along and with that are named here. And I'll try to call out their contributions as we go on. But in a paper that was published in Cell that was first authored by Moshe when he was a postdoc in the Hacohen lab, Nir had identified that there are different flavors of CD8 T cells. Not all CD8 T cells are created equal. And ultimately, some are good and bad, labeled here G and B, essentially meaning that the good ones are more associated with clinical response to immunotherapy. And I should, in case I breezed past this is single cell RNA sequencing, melanoma patient tumors. And then ultimately, we were able to clinically annotate them. So we had 31 non-responding tumors and 17 responding tumors and 31 non-responding tumors. And then identify which ones are associated with clinical response and resistance. And ultimately, we were able to identify what the gene programs were associated with those different T cell states, as they refer. But ultimately, the good T cells were characterized by high levels of this transcription factor, TCF7, and the quote unquote bad T cells had high levels of these T cell exhaustion markers, including CD39, TIM3, and as I'll show you in a moment, CD38. Our contribution to this effort is that single cell RNA sequencing is ultimately a descriptive tool. It's hypothesis generating. Ultimately, this is the discovery phase of any investigation, and ultimately there needs to be validation. And while in vivo studies are great, they're not everything. So we wanted to do some manipulation of the immune microenvironment. And ultimately what we showed was the following, which I'll try to go through relatively briefly since, again, this is published. But if we go to one of these MDOTS models, here the CT26, which is partially responsive to PD-1 blockade. So we take the tumor from the immune-competent mouse. We know there's a certain proportion of T cells in the tumor microenvironment. We treat with PD-1, we get a little bit of response. So what we did is we used flow sorting to enrich for either the cells that have low expression of CD39 and TIM3. These are the good ones, the ones associated with immunotherapy response, or we sort those that are double expressors or double positive, DP here, for CD39 and TIM3. These are the more exhausted ones. And then we enrich 100-fold. And so for this particular column here, we've got our standard MDOTs, but we've augmented them with additional tumor infiltrating immune cells that are the double negative variety, the quote unquote good ones. And sure enough, they actually enhance the sensitivity to PD-1 blockade, demonstrated here by the increase in red or dead cells. Contrast that to the the double positive cells, the ones that are the bad guys here, that are double positive for CD39 TIM3. Adding those not only didn't enhance, it actually blunted the efficacy of the PD-1 blockade compared to if we did nothing at all. If we had equal proportions of them, it's a tie. It's as if we added nothing to the cells or to the MDOTs here. So the proportion of those different populations we think is vital to determine whether or not a given tumor, and certainly in the MDOTs model, a given MDOT specimen is going to respond or not. So this gets to the balance of CD8 T cell states and response and resistance. So if we go back to the original data, what are some cell surface targets, let's say, something that could be targetable by a monoclonal antibody that are maybe enriched on the bad guys that either could be depleted or blunted or otherwise targeted? So sure enough, CD38 was the thing that scored as the top statistical, enriched in the non-responders. So here, purple means non-responder clinically, and this is just subclustering on the CD8 T cells, and this is CD38. And in contrast, the transcription factor TCF7 is more enriched on the East Coast here. We've got CD38 more on the West Coast. And if we look at the clinically annotated specimens, those from responders and non-responders, we see the proportion of CD38 expressing cells is significantly higher on the CD8 T cells from the non-responders. It's one of the most predictive features that's been identified using this data set with a very high area under the curve for the ROC curve. And if we look in an independent data set that has not yet been published, we actually see the exact same characteristics, that there's far more CD38 expressing CD8 T cells in the patients that do not respond to immune checkpoint blockade. Clinically, Karen Yitzhak and Ophir Shore did the analysis on the previous data, and Jackie Yang, who is in Manolis Kellis's lab at MIT, has her own independent group at Rutgers, and Javier Boland, one of my surgical colleagues here at MGH, were responsible for the new data set. And if we look at whether or not baseline samples or post-treatment samples are examined, we can see that it's statistically significant in both of those situations. Keeping my eye on the clock, there's a lot of clinical data here that's supporting the target of CD38 that I'm going to try to go through relatively quickly, so there's time for some of the biology at the end. But suffice to say, we've sliced and diced the data a variety of different ways, because CD38 is a molecule that can be expressed on a number of different cell types. Clinically, CD38 is targeted multiple myeloma as the drug daratumumab. And so we know it's expressed highly on B cells and related immune cells. So we wanted to see, using all the different immune cell, single cell RNA sequencing data, was CD38 expression associated with response or resistance more broadly? Was this unique to T cells? And if so, was it only the exhausted T cells? So the long story short is that the most predictive was if we just looked at CD38 on the exhausted T cells, and CD38 in these other populations had some signal, but it was greatly diminished compared to the exhausted T cell population. So what about peripheral blood? Do we really need to get into the tumor to measure this at all? Can't we just do a blood draw to understand this? And sure enough, we actually can. It's small percentages of cells compared to what's in the tumor, but you can actually look, and there's a pretty wide variety, or range, I should say, not variety. But there, if you look in CD8 T cells using mass cytometry, and this is data that was generated in collaboration with Tycho Bio out of Salt Lake City, we can see that there's more CD38 expression in the CD8 T cells from non-responders. However, you look at baseline pre-immunotherapy, it's not statistically significant. It's only after treatment do we see these shifts. And this will become, I think, a little bit more clear why we think this is the case in a few slides from now. Interestingly enough, if we look at those on-treatment specimens, we can actually, since the anti-PD-1 antibodies that are used clinically, like pembrolizumab and nivolumab or IgG4 antibodies, we can do an anti-IgG4 and actually mark those T cells that are already pre-coded with anti-PD-1. And in those CD8 T cells that already have an anti-PD-1 antibody bound to them, there's higher CD38 expression in the non-responders, which is a pretty nice way of saying that the PD-1 didn't work perfectly and it's actually upregulating CD38. So pivoting back to mouse data for just a moment, one of the things that we've done is partner with a couple groups that have orthogonal data sets that we thought might be useful to understand the biology around CD38. And again, this will come back to MDOTs and PDOTs in a moment, but one of the things I think is important anytime you're developing a new target or evaluating a new target is to kind of rely on all the different tools that are available to understand exactly what the relevant biology might be and where it might be taking place. So this particular data set, Lloyd published in Nature in early 2023 when he was a postdoc in Vijay Kuttru's lab. And essentially what he had done was do single cell RNA sequencing of the B16 murine melanoma model at early time points, day 7, halfway time points, day 10, and then late time points, essentially being these ones are the more immunotherapy sensitive versions. If you let the tumors get bigger, they lose their capacity to respond to immunotherapy as time goes on. Eventually they get so big that mice need to be sacrificed. And these later tumors get very, very resistant and grow very, very aggressively. But in addition to looking at the tumor infiltrating immune cells, Lloyd also took down the tumor draining lymph nodes and the non-tumor draining lymph nodes to examine whether the key immune cell populations were truly restricted to the tumor or not. And so this is the UMAP showing the different in UMAP space, the location of the immune cells from tumors, draining lymph nodes, non-draining lymph nodes at the different time points. And this was what we learned about CD38, is that CD38 was uniquely upregulated in the tumor infiltrating immune cells, but was not detected at all in the tumor draining lymph node or the normal draining lymph node. And if we look at this over time and we contrast that with the transcription factor associated with the good T cells, TCF7, we can see high levels of the good guys in the normal lymph nodes, the draining lymph nodes. And if we look at these early time points, again, over here where the tumors are just starting to grow and they're immunotherapy responsive, we see a little bit of CD38 and a little bit of TCF7, but as the tumors get bigger and bigger, we lose the transcription factor TCF7 and we get higher levels of CD38. So there does appear to be a switch, a transition here. And these UMAPs on the bottom are just meant to show in the UMAP space that the CD38 expressing cells are coming from the tumor, showing essentially what I showed you on those dot plots above. And as the peripheral blood profiling data would suggest, if we treat mice in vivo with an anti-PD-1 antibody, we see the same sort of response. So this is, again, the B16 model. I'm going to move through this quickly. But if we look in single-cell RNA sequencing data by this galaxy plot, you can see after treatment with an anti-PD-1 antibody, there's an increase of these both early effector CD8 cells and the late, more terminal effector CD8 T cells. And the terminal effector is where most of these CD38 expressing cells come from. In these TRAX plots, you can see the shift that is mostly ends up being statistically significant. The CD38 score, if we look at the terminal effector subset of T cells. So back to the human for a moment, we go back to our original patient-derived melanoma, immunotherapy responsive, sensitive, all the data together. And we look and see what other genes are kind of co-expressed with CD38. Again, all of this is going to come back to PDOTs, I promise. But one of the things we observed is that co-expressed with these cells that have high levels of CD38 are PD1, TIM3, CD39, CTLA4, LAG3. It's all the other exhaustion markers that have been targeted clinically, and it's the other populations, generally speaking, that have those markers of a more naive or stem memory state here. We looked at a correlation or co-expression deviation proportion analysis, courtesy of David Liu and Jackie Yang, and indeed they all kind of co-express with each other and kind of are inversely correlated with TCF7 expression. And if we look at the differential gene expression using the single cell RNA-seq data, we see essentially the same thing. There's a lot of genes associated with T cell dysfunction, exhaustion, but also immune activation that go along on the right side here. And again, those that are differentially expressed in the cells lacking CD38 expression include the transcription factor TCF7, recurring theme. We can actually take a mixed population of T cells and sort them using flow sorting and take the portion that are CD38 high versus CD38 low and see that the ones that have high CD38 have more exhaustion markers and lower TCF7. I'm beating the horse here, so I'm going to keep on moving here. So we developed an in vitro T cell exhaustion protocol to see if we can take a more naive T cell population and beat it up so it approximates what we see in tumors, which is not entirely straightforward. And there's a variety of different protocols to do this. We've done a repetitive T cell receptor stimulation model using anti-CD38 and anti-CD3 and CD28 beads. And eventually the cells over time with this kind of chronic repetitive stim start proliferating less. They start upregulating CD38, CD39, and TIM3, and they become less effective killers as a consequence. So they meet all the basic criteria for us labeling them dysfunctional or exhausted. And we can actually go in and introduce a CRISPR guide against CD38 up front and can essentially prevent that from happening and associate with that process is higher TCF7 and improved cytotoxicity. So this is all the background for this particular slide. So if I could show you one slide to pay attention to, I think this is the one. So this is all the lead up as to the work that went in behind the scenes to nominate the target, to validate the target in vitro systems and using computational tools with existing data sets. But this is the phase -1 clinical trial we wanted to do to prove whether or not blocking CD38 would actually overcome resistance or enhance sensitivity to anti-PD-1 treatment. So this is a cohort of 30 patients, mostly melanoma. We generated our PDOTs. We have an untreated control, anti-PD-1, pembrolizumab, anti-CD38, daratumumab, or the combination of these two. We embed them in the microfluidic device, we treat them, and then they are grown from 5 to 7 days, and then we do our end of treatment image-based viability assessment. This particular cohort was very, very PD-1 resistant because this was mostly melanoma and patients that were pre-treated with immunotherapy and did not respond. And this was done deliberately to try to enhance the signal for augmentation of response in the most clinically relevant population. CD38 on its own had some single agent activity, not tons, but it did have some. But the combination was really mind-bogglingly good. We had greater than 50% of our patient specimens cross this threshold that we set of 30% reduction in PDOTs viability compared to baseline. And these are our different tumor types. Again, mostly cutaneous melanoma or non-cutaneous melanoma, and Merkel cell is a rare skin cancer that is also PD-1 sensitive that was included in a couple GI cancers just for kind of evaluation of other tumor types. But we go from, a response rate of about 3% to over 50% by the addition of an anti-CD38 antibody. We had collected single cell suspensions from these same tumors and run flow cytometry and condensing a whole lot of data in collaboration with Arnav Mehta, who's co-mentored by Eric Lander and Nir Cohen, and Sam Wright, an associate computational biologist working with Arnav. We had run to see linear regression multivariate analysis to see what features in the native tumor immune microenvironment were most closely associated with ex vivo sensitivity to dual PD-1 CD38 blockade. And the things that we found were CD8 T cells expressing PD-1 and CD38, which all was very nice and internally consistent with what we observed. You needed a certain amount of PD-1 expression and CD38 expression on your CD8 T cells in order to see this killing. But we could do this all outside of a living organism, which is pretty wild. So to show a couple of the greatest hits from our PDOTs data, this was PDOTs from one particular patient that was immunotherapy resistant, had little activity to any of the single agent, had a very nice reduction in cell viability with the combination. This was cutaneous melanoma. Mucosal melanoma is a rare subtype of melanoma that is far less responsive to immune checkpoint blockade. We saw essentially no activity to pembrolizumab, some single agent activity with daratumumab, and really nice synergy with the two. This is what it looks like at the individual spheroid level under the microscope. And we can actually do some limited multiplex immunofluorescence staining to find the immune cells that are actually coded with CD38 and PD-1 on chip in the microfluidic device. Going back to the mouse models, again, we wanted to make sure that whatever we're observing in our human system, we can observe in mice and vice versa. So we took, M-dots from the B16 ova model, which is partially sensitive only because this is expressing a xenoantigen, the ova or ovalbumin here. And here we were deliberately looking for later tumors. If you recall the data I showed not that long ago, showing that as the tumors get bigger, the CD38 goes up and the TCF7 goes down. We're now looking at day 10 or day 14. So these are the tumors as a starting point that have high CD38 and low TCF7. So there's essentially no response to PD-1 blockade here, modest response to CD38 blockade, and a very nice synergy to the combination. The CT26 GFP model, again, minimally sensitive to PD-1, very nice synergy with the combination. And this is what it looks like under the microscope, again, with the increase of red indicating the dead cells here with the combination treatment. So we toggled back to our single cell RNA sequencing data to try to teach us how this was actually happening. What was the mechanism whereby CD38 was modifying this? And using gene set enrichment analysis, we observed a couple things. The top scoring gene set actually associated with being a CD38 high T cell was actually oxidative phosphorylation, which was initially quite stunning to us. And if we did this comparing both the human single cell RNA sequencing data and our murine single cell RNA sequencing data, we saw that they actually seemed to score reasonably well. This wasn't a uniquely human or uniquely murine process. And in addition to that, we saw interferon response associated with being a CD38 high T cell. So the first thing we did is we actually treated the isolated T cells in vitro. This is not PDOTs or MDOTs, just good old-fashioned in vitro work. We treated T cells with interferon beta. Long story short, we looked at other interferons, and this is a type 1 interferon unique response. Interferon beta is sufficient to induce levels of CD38. And if you activate the T cell receptor at the same time, you can further augment this. Interestingly enough, interferon beta doesn't do as much to the expression of PD-1 and TIM3, but it does augment the expression in the setting of T cell receptor stimulation. And under both, it's sufficient to decrease the expression of TCF7. If we go back to the mitochondrial angle here, there's actually some existing literature suggesting an interplay between CD38 and mitochondrial function as a consequence of CD38's role. And it's an ectoenzyme that degrades nicotinamide adenine dinucleotide, or NAD, key bioenergetic metabolite. So we went and looked, and we found that in the T cell population here, there's actually a marked increase, surprisingly increase in mitochondrial mass and an increase in mitochondrial membrane potential, which was initially quite stunning. Others have actually shown that in some of these exhausted dysfunctional T cells, there's some sort of problem with mitophagy and an accumulation of aberrant, odd mitochondria. And again, we haven't delved into this too much, but I welcome the feedback from the mitochondriacs on the call, Dr. Latai included. And interestingly enough, when we CRISPR out CD38, we see the opposite. We see the mitochondrial mass with mitotracker green, mitochondrial membrane potential with mitotracker red, which is essentially like tetramethylrhodamine, going down. And if we do some seahorse assays to look at oxygen consumption rate, we can see by getting rid of CD38, we're able to increase this particular kind of peak or maximal respiratory capacity. And we can do this both by knocking out CD38 or using a small molecule inhibitor of the enzymatic function of CD38, suggesting again that there's kind of improved T cell fitness under these conditions of enhanced metabolic stress that maybe goes hand in hand with being an exhausted T cell and facing persistent antigen exposure. So in collaboration with Nama Kanarek at Boston Children's, we had done some metabolomics by LCMS. I'll try to summarize quickly, but as one would probably expect, by blocking this enzyme here, CD38, we're getting an accumulation or increase in NAD, but we're also funneling things towards NADP. And we're expectedly seeing a decrease in the product of CD38, which is ADP ribose or ADPR here. And when that happens, again, all in vitro here, we're seeing a decrease in the markers of exhaustion, increase of TCF7, the good guy transcription factor. So it's all seeming, for the most part, like a nice, tidy package here. So one question we had, going back to the therapeutic of choice, is, was there something unique about the activity of the antibody against CD38 we had used for our PDOTS model, daratumumab, which is an IgG1, which could potentially engage antibody-directed cellular cytotoxicity or do other things with macrophages that might facilitate. So we went back to our small molecule inhibitor that worked so well in vitro, and we could actually demonstrate in our MDOTS models that an anti-PD-1 antibody and a small molecule inhibitor of CD38 were enough to synergize. Same in the CT26 model. And going one step further, we could do kind of a metabolic bypass here, not using a small molecule inhibitor. For comparator, we've got the PD-1 anti-CD38 here, but here we're using nicotinamide ribosides. This is a way of essentially boosting cellular NAD levels, has very little activity on its own, modest amounts, but in combination with PD-1 was pretty effective in this particular model system. And again, we've done a separate cohort of PDOTs compared pembrolizumab with the small molecule inhibitor CD38, which is 78C, the small molecule inhibitor for those interested in the combination, which also worked reasonably well. So to sum up, CD38 high CD8 T cells are associated with immunotherapy resistance. The single cell data seems to bear this out pretty clearly. CD38 high CD8 T cells are dysfunctional. Targeting CD38 restores TCF7 expression, again, the transcription factor associated with good guy CD8 T cells and T cell proliferation. Targeting CD38 overcomes immunotherapy resistance. Again, I think best exemplified not just with our MDOTS models, but in particular our PDOTS models. The living tumor biopsies, again, largely from patients melanoma patients clinically resistant to immunotherapy, we can go from like no response to PD-1 with this combination strategy going to over 50%. And then NAD boosting, simply increasing the cellular NAD pools, phenocopy CD38 inhibition or blockade, and in some instances may actually augment it. We have a preprint up for those that are interested in reading a little bit more. It's out for review now, 31 days and counting. So hopefully we'll hear back soon. But others have taken note. There's some publications from earlier this year showing that inhibiting CD38 enzymatic function actually can make CAR T cells less prone to dysfunction, and it ultimately it's feeding into CAR T metabolism. And this paper, getting into a little bit more the mechanism downstream of CD38, is invoking yet another process beyond NAD metabolism, saying the product of CD38 ADP ribose is signaling through calcium, which was previously known, but not in this particular context. So it could be that CD38 is regulating NAD, which is promoting, we think, TCF7 expression, potentially be a NAD requiring epigenetic regulators like sirtuins, and then also this calcium-mediated signaling downstream of ADPR. This might be a kind of a dual toggle that the T cells use to kind of shift metabolic activity and gene expression at the same time. And going back to the things that we ultimately care deeply about, this falls very squarely in category 2, developing rational combination approaches. We're actively in discussions to bring this forward in a clinical trial using either daratumumab or isatuximab, the 2 FDA-approved anti-CD38 antibodies. And again, ultimately, we'd like to think of this as a precision tool, but there may be opportunities to take clinical trials that will target a broader population and do the correlative studies to demonstrate that a functional approach is going to be more meaningful than any of the static biomarkers. So hopefully that we can take these instances where there's good preclinical data, make it good clinical data, and then ultimately feed that back to augment folks paying attention to functional precision medicine. And this is to say that this is currently being done as part of a broader effort to integrate these static analyses, whether it's spatial, single cell, ATAC-seq, along with kind of functional drug sensitivity or perturbation studies to try to use the best of what's around to try to nominate the best targets and then actually test them in the most meaningful model systems around. There's tons of people to thank. Again, I want to leave enough time for questions. I kind of acknowledge a couple folks along the way, but we have a lot of great funding support, wonderful collaborators, very amazing clinical support staff, and of course the patients, their families, for making the donation of their tumor tissue, without which we couldn't do what we do. So I will stop there and take any questions.
Great. Thanks a lot, Russ. That was, I'm glad you took the opportunity. Obviously I wasn't very familiar with any of the unpublished data, even though, you know, for many years I was aware of the PDOT work that you guys started in Dave's lab. As almost always I do, I neglected to ask people, if you have questions, you can either put your hand up and ask them in person or put them in the chat. So while we're waiting for that, I have a couple questions. I think you gave, so with regard to like functional precision medicine, you gave a really very good example of using living patient tissue in what I might call an investigative or discovery aim. And I think that's
Yes.
a very important use of it. I think there has been, as we all know, a very strong reliance on using genetic mutations to guide this sort of discovery over the last 2 decades. But I think any realist would have to acknowledge that we've probably identified most of the most targetable genetic alterations in cancer at this point. And we've certainly identified that maybe there's still a biochemical task or a pharmaceutical task to target some of them, and we still have failed to do. But the idea that there are great targets existing out there that will help a lot of people is probably incorrect. And in spite of that, I think we do see people continue to try to work that field.
Right.
But I wanted to get back to kind of your initial introduction, where you were talking about using it as a predictive biomarker. Now, I actually looked like the performance was okay for that, but I am not aware of any trials using anything like what you do or anything like it to assign patient therapy. or to predict response. So let me ask you, what do you see as the major obstacles to making that a reality? Why isn't it happening? It seems like I'm always asked, are there any predictive biomarkers for immuno-oncology therapies? I go, well, I think there are. And yet people say it's a desperate need, but pharmaceutical companies aren't using them.
100%. So I can say on the preclinical side, no one wants to hear that their baby is ugly. So no one wants to hear that if you test their drug in PDOTs and it doesn't work, that it doesn't work. So there's, I think, general opposition to the notion of identifying those targets early and saying, oh, I think this one's a dud. So that's that part. But going all the way to what you were talking about earlier, if we've got drugs in the clinic and they work some of the time, why can't we do functional precision approaches? The biggest barrier currently is that there is no clinical indication for patients to have an excisional biopsy pre-treatment. The only patients that do that are volunteering, and it's not something that pathologists bill for, or excuse me, or the surgeons bill for. It's purely done on a research basis because there's no need.
You said excisional biopsy. Would you settle for a needle biopsy? Not
enough cells. Core needle biopsy will be great for single cell RNA sequencing. You can do your 10x and have 8 to 10,000 viable cells. That's the number of cells we load in a single microfluidic device, and we do triplicates for every treatment arm. So not for us, unfortunately. We tried this a number of years ago in Dave's lab.
So how many cells do you need to sort of do a run of what you want to do?
So we aim to load anywhere between 10 and 50,000 cells in 10 microliters of collagen in the microfluidic device. And so usually this requires an excisional biopsy. Amazingly, they don't need to be huge. People will often ask, well, how big does it need to be? And my answer is, it just needs to be not dead. It needs to be, because a lot of the larger tumors will actually get very, very necrotic, and you end up with small portions of viable tumor on the periphery and this kind of liquefied necrotic nastiness on the inside. So we've taken teeny tiny little tumors, Merkel cell carcinoma, melanomas, and they yield enough material for us to test 8, 10 different treatment arms in triplicate. So the size isn't always the issue. In a pre-treatment therapy naive setting, we probably could get away with smaller specimens. The instance is that, number one, melanoma often has patients that will have metastatic lesions in lymph nodes that are palpable, clinically accessible, or subcutaneous dermal or epidermal metastases. And so they'll volunteer in clinic or we'll invite them to participate in clinical research. But there is, as of yet, no opportunity to do that. The two opportunities, and we'll see if NIH bites on this, because this is the grant that they'll be reviewing tomorrow. The resubmission was the proposition of expanding the context of use for PDOTs for 2 clinical scenarios for non-interventional co-clinical trials. The first being melanoma that is metastasized to the brain, because often that's a very dramatic emergent process. The patient has melanoma, goes to the brain, they have neurosurgery, a dominant lesion is removed, but almost invariably they've got widespread distant metastatic disease. But that's a large amount of tumor that can be tested upfront for assessment. All it takes is coordination with the neurosurgeons. And those patients are all getting dual PD-1 CTLA-4 blockade. And the response rate is 57%. That's the Checkmate 204 study. So you've got a nice response rate, clean cohort, everybody's treatment naive. It would be ideal. We just need the funding to do it and to support all of the other ancillary collaborative folks to make it happen. The second, which there's huge enthusiasm, and Christian Blanc presented at ASCO this year the results of the NADINA study that essentially showed that neoadjuvant preoperative immunotherapy is 100% hands down better than doing upfront surgery and then adjuvant postoperative preventative immunotherapy. And the pathologic complete response rate is near 60%, meaning that patients get 2 doses 3 weeks apart of combination PD-1 CTLA-4 blockade, slightly different dosing regimen than for the advanced metastatic setting. But those patients have a 70% reduction in the risk of melanoma recurrence compared doing postoperative immunotherapy. Again, high response rate, easy to benchmark, 6-week window of time. You just need a reason to sample a little extra tissue up front that most patients don't sign up for unless it's a clinical trial and it's mandated. But again, our overall enterprise is moving in this direction. There's lots of interest, excitement, enthusiasm around the neoadjuvant approach. So those 2 instances, I think, will be our chance to have real impact. We've now, over the last 5+ years, we were just tallying this, we've done over 400 PDOT specimens, over 200 melanoma PDOT specimens, but the response rate to PD-1 in our cohort is 10%, because most of the specimens we get are from patients that have immunotherapy resistance and they're symptomatic, which is why they're having surgery in the first place. So to answer that question of prediction, we have to go upfront, treatment naive, and it has to be ideally a clean cohort where there's a clinical indication and potentially the capacity to collect excisional tumor specimens. And I think those are the two best cases. We'll see if NIH agrees.
So I'll say, like, to me, those sounds like I sound like very good context to do your testing. And if you get funded and you do those studies, you'll have a great opportunity to show that it works. And I'll just tell you that, and probably everyone is familiar that if you do that and you show that it works, there is still going to be the existing hurdle of convincing pharma that they should want to use this. And that has all sorts of financial and logistical implications that we don't need to get into. But like, I agree, that's the sort of study that we need to do. Let us know. We're happy to publicize it once you get that out. But I see there is a question in the chat from João Montero. I think this is always an important question for people doing ex vivo models like this. What is the sort of take rate or success rate of your PDOT? So like, if you get 100 melanoma samples, how often can you successfully raise a PDOT?
So we're actually preparing this data now, Johan. So we'll hopefully have it out for the world. But initially, my kind of back of the envelope calculation was we were in the 80 to 90% success rate range. And then we're actually doing a deep dive on kind of melanoma and non-melanoma tumors right now. And in melanoma, it was lower than expected. It was around 70%. And so we looked at our failures now. And interestingly enough, for some of those tumors that failed to yield spheroids amenable to testing, those patients had prior therapy. And if we look at the pathologist report, there was no viable tumor. So it's actually an interesting positive for a negative, is that the inability to generate PDOTs is actually probably predictive of absence of tumor cells. And so in many of those instances over the last year have been patients receiving neoadjuvant upfront immunotherapy, those 2 cycles of immunotherapy where they're getting the pathologist doing a deep dive, multiple sections, and say, no viable tumor detected. We don't get any PDOTs out of those. So that's the flip side. But I'd say overall, just by the numbers, it was like 70%.
So there's a question that I think you sort of hinted at from Etienne Laurent, where he's asking, how do you make sure there is actual cancer in the specimen before you put it on the chip? Or do you?
Currently, we don't. There's the Gestalt approach right now, which is, does it form spheroid? So if you take tumor and normal adjacent tissue and you try to, our standard protocol for dissociating tumor, there's something about having cancer cells present. They will maintain some of those interactions with those other cells, where if it's normal tissue, tonsil, pancreas, whatever, it just kind of disperses into single cells. So we usually know by Gestalt whether or not it's fibrotic, necrotic, if there's any viable stuff, but sometimes we'll kind of load the leftovers and see what we can see. It's hard to do that on the actual multicellular tumor aggregate. So most of the baseline examination we do in terms of the flow cytometry, we actually do as a post hoc analysis. We'll do all our tumor specimens, and then we'll thaw the specimen, do the staining, and then do the flow all at once on the back end to reduce batch variability from batch to batch or assay to assay. So oftentimes we don't know. And sometimes that you can use that to your advantage and say, listen, this is what we observed. And then on the back end, we observed that there was this proportion or there was no tumor cells. And that explains, why it did or did not have a given effect.
Okay, great. Well, I appreciate, I think we've reached the top of the hour. If you have any further questions for Russ, reach out to him. His email is pretty publicly available. Thanks, Russ. That's really interested. Keep us in touch with how this progresses.
Will do. Thanks, Tony. Thanks, everyone. Appreciate the opportunity.
Take care, everyone. See you next month.
Bye.